Hello everyone, my name is Maria Laura Polo and my today's presentation is entitled SARS-CoV-2 Immunity in COVID-19 Convalescent Individuals Living with HIV, Bulk Immune Profiling and SARS-CoV-2 Specific Humoral and Cellular Immune Responses. As an introduction, we can mention that despite significant advances have been made, still is not completely understood whether people living with HIV and AIDS can be more likely to present severe manifestations of COVID-19. Moreover, SARS-CoV-2 specific immune response features remain to be fully elucidated in this population. In this regard, the aim of this work was to study the impact of HIV over the immune profile of lymphocyte populations, as well as the humoral and cellular response secondary to SARS-CoV-2 in people living with HIV recovered from COVID-19. Since the beginning of the pandemic, the Argentinian Biobank of Infectious Diseases started collecting blood donations from people with COVID-19 diagnosis. Each sample consisted of 40 milliliters of whole blood, which was processed to obtain serum, plasma, and PBMCs. In this work, plasma and PBMC samples from 21 people living with HIV on ART and 21 HIV-negative participants with COVID-19 diagnosis were studied evaluating SARS-CoV-2 specific IgG levels and titers, IgG neutralization capacity, SARS-CoV-2 specific T-cell response, and frequency and phenotype of bulk T, B, and NK cells. These are the participants' characteristics. People living with HIV and HIV-negative individuals were matched by age and by days since symptoms onset. People living with HIV showed no detectable viral load and preserved CD4 T-cell count. As a surrogate marker of disease severity, we quantified the number of symptoms displayed by the individuals, finding no differences between the two groups. Also, the distribution of clinical manifestations was similar among the individuals. All of them presented mild disease with the typical manifestations of COVID-19. Regarding SARS-CoV-2 specific humoral response, neither the frequency of seroconverted individuals nor the levels of IgG differed between the two groups. It's important to mention that we found a positive significant correlation between the CD4 T-cell counts and both the IgG titers and neutralization activity of these antibodies, suggesting that the more preserved the immune system of people living with HIV is, the more efficiently the humoral response can be mounted. By means of flow cytometry, we studied the frequency of lymphocyte B cells and antibody-secreting cells. Here you can see representative examples of these cytometries for both groups. Compared with people living with HIV, HIV-negative individuals displayed increased frequency of antibody-secreting cells. Also, there were no differences in terms of CD4 and CD8 T-cell subsets, but expression of PD-1 and expression of HLA-DR were upregulated in CD4 and CD8 T-cells from people living with HIV. Additionally, the distributions of T helper cells and Tregs were similar between the two groups. On the other hand, the frequency of T follicular helper cells as well as the CXCR3 expressing follicular helper cells were upregulated in people living with HIV. Within this group, when we evaluated a larger number of samples, we found a negative correlation between the IgG titer and the frequency of follicular helper cells. Regarding NK cell compartment, percentages of these cells were comparable between the two groups. However, NK cells from people living with HIV displayed a more activated phenotype showing increased expression of CD25, CD95, DR, and NKp46. Finally, we evaluated SARS-CoV-2 specific T-cell immunity by measuring interferon gamma production upon stimulation with spike and N proteins, RBD as a fraction of spike, and S and N peptide pools. As you can see, interferon production following stimulation with spike, N and S pool was higher in HIV-negative individuals compared to people living with HIV. To sum up, exacerbation of COVID severity was not observed in this group of people living with HIV with preserved CD4 T-cell count. As previously reported, people living with HIV showed an immune profile with enhanced activation and exhaustion, as well as increased proportions of follicular helper cells. On the other hand, people living with HIV could elicit SARS-CoV-2 specific cellular response, but in a lower magnitude than HIV-negative individuals. Finally, in this population, preserved CD4 T-cell count emerged as a key factor to achieve better antibody responses with higher neutralization capacity. Thank you for your attention, and we would like to thank all the donors and collaborators to this project.